Hello, Mr. Almond here, and this is your Almond Cast on the kidney. The kidney is a part of a human anatomy. We will study a lot of anatomy and physiology over the next 2 years. While anatomy is something that may be more familiar, people could have a vague notion of what the kidney is and where it's located. This, however, isn't really the significant part of understanding parts of the body. Rather, it's the processes that those parts undertake in order to keep you alive. In this unit, you learn that osmosis is the process by which water moves from areas of lower to higher solute concentration. It is that movement of water that is going to be very important in allowing your body to be able to excrete waste. In this particular video, I want to take you through the physiological processes by which the kidney goes about the production of urine, which ultimately allows for you to be able to maintain the amounts of salt balance in your body. Later in the video, we'll compare the anatomy of the kangaroo rat versus the human in terms of what's happening inside its kidney. That can be explained as you consider the differing environments in which they live. A kangaroo rat which lives in the desert is going to get most of its water from the chemical byproduct of breaking down food. It doesn't drink at all because there's no water available where it lives. As a result, it produces very little urine, and most of its water loss actually comes through evaporation through its skin. On the other hand, humans live in areas where there's plentiful water, and so we ingest most of the liquid in our bodies, and consequently we also release or excrete much of that water, not from evaporation, but through urine. Animals need to be able to regulate their internal osmolarity, which you can think of essentially as the breakdown between the concentration of salt in the body versus the concentration of water in the body. And that ratio needs to be maintained. So, in the production of urine, an animal can produce more or less concentrated urine. More concentrated urine is going to have more solutes dissolved in it. Less concentrated urine is going to have less solutes in it. Secondly, by controlling the amount of urine produced, an animal can produce more or less urine based upon its internal conditions. Finally, the internal conditions can be affected by the environment. So behavior, producing hunger, particularly a hunger for salt, or more commonly, producing thirst so that the behavioral response will be to take in more water as a way to be able to balance out in a case when water is deficient. All animals are going to have a very specialized system of tubules that are going to move materials either into or out of the blood. In many cases, there can be much more complex types of ductwork and more complex types of species-specific parts that will allow that organism to be able to survive in its particular environment. Being able to cry out salt isn't something that we need to do in order to maintain our osmolarity. But if you live in a place that's surrounded only with seawater, then that would be a necessary adaptation for you to have. A second reason, other than regulating osmolarity, why it is that we have an excretory system and a kidney in particular, is to be able to deal with a very particular type of waste product produced in our cells. Metabolism is the sum of all the chemical reactions that happen within our cells, the most important of which is cell respiration. And in cell respiration, there are going to be waste products produced as proteins and nucleic acids are built up and broken down. Now, once those things are broken down, something needs to happen to them because they are actually toxic. Now, some organisms produce ammonia, or as in humans, we could have ammonia as a byproduct, which will then be converted into one of two other forms in land animals. Urea, which is found in humans, or uric acid, which is more common in things like birds. For mammals, which we'll focus on through the rest of this video, animals produce urea, which you can see the structure of in the middle. Urea is a relatively small but polar molecule containing two atoms of nitrogen. This nitrogen-containing molecule isn't very toxic, but if it builds up, then it is going to become toxic within cells. Therefore, it needs to get out. It does dissolve in water, but does need to move by facilitated diffusion across the plasma membrane. So we get to the excretory system. The four terms here are a brief outline of the major processes that we see happening within the excretory systems of any organism. First, excretory systems will begin with the process of ultrafiltration. Ultrafiltration is going to be when there's initial movement of fluid and solutes from the body into the excretory system. So within humans, those fluids are coming from the blood, and we're going to have a filtration process by which water and anything dissolved in that water is going to move out of the blood and into the tubule, and it becomes now a substance that we call the filtrate. Secondly, there's going to be reabsorption. Reabsorption is going to be when some of that water and any desirable solutes, things like sugar, salts that are found in low quantities in the blood, amino acids, those things that may initially have moved into the filtrate but are needed to maintain life, those things are going to be reabsorbed back into the blood. Third, secretion. The process of filtration isn't going to capture all of the water or all of the waste that's found within the blood. So therefore, third, having a special process by which some of those excess waste solutes, things like urea and excess sodium, some way to be able to get them in very large concentrations into the filtrate so that the blood is rid of them entirely. And finally, excretion. Excretion is when that filtrate is then going to be expelled from the body, being sent along to the bladder and leaving through the urethra as urine. So as we look specifically at the vertebrate excretory system, using the humans as a more specific model, we have four main parts to the human excretory system. It starts with the kidneys. You have two kidneys located more to the dorsal side, meaning closer to the back, just above the waist. This is going to be where the filtrate is collected and the urine is produced. The ureters are going to be the tubules that are going to carry the filtrate down from the kidney into the bladder. The bladder is where urine will be stored until ultimately it'll be excreted from the body through the final tubule called the urethra. Now within the kidney structure is going to have two main parts. We have the cortex. The cortex is found along the whole exterior of the kidney. Then we have the medulla. Now the nephron is the functional unit of the kidney. There are about 1 million of these located within the organ. And so if you understand what one nephron does, you understand what the organ as a whole is doing. The nephron extends across both of those main two parts of the kidney. The structures at the top of this particular image are going to be located in the cortex, while the part looping down below is going to extend down into the medulla. The process of being able to filter the blood and excrete waste as urine is going to begin with ultrafiltration, then the process of reabsorption as it moves down. The process of secretion is going to continue along with reabsorption throughout this loop, and then secretion is going to complete as it moves back up to the top, back into the cortex. There's some important anatomical parts here that you need to be familiar with, as you do need to be able to annotate the structure and function of the nephron. The nephron begins at the top with a structure that's not a part of the nephron but is associated with it, called the glomerulus. The glomerulus is going to be a ball of capillaries, pictured here as a small net. As a small net within a larger structure, the first part of the kidney nephron, which is called the Bowmans Capsule. this is the place where ultrafiltration occurs, where water and any dissolved solutes are passing out of the blood and are being collected by the Bowmans capsule to be able to start moving through the tubules of the kidney nephron. Back at the proximal tubule, this is where reabsorption is going to begin, and many of the nutrients, as well as some of the water and salts, are going to be brought back into the blood. In the loop of Henle, which extends down at the end of the proximal tubule, extending down into the medulla, this is going to be where more water will be reabsorbed, as well as salt. And then some of that salt actually may be, depending on its concentration in the blood, may be then secreted along with urea as we get to the ascending branch of the loop of Henle. The distal tubule is going to be what collects from now this increasingly more concentrated urine. The final reabsorption of water, salts, and urea will occur there, as well as secretion of any additional solutes if still present in the blood. I want to show you a couple of diagrams here that are trying to give you a schematic by which to be able to appreciate exactly what's happening and when. Now, first of all, this is in the cells unit because we have an application here of active and passive transport, as well as the importance of osmolarity in determining when and if solutes are going to pass across the membrane from the blood into the kidney or vice versa. First of all, we have ultrafiltration, which is mostly a physical process. So those molecules that are smaller are going to pass by diffusion, passive diffusion, from the blood and into the filtrate. We can see an application of active transport most clearly in the transportation of nutrients from the filtrate back into the blood, because those nutrients, things like glucose, are necessary for cells, and it would be a waste to leave them in the filtrate and ultimately excrete them as part of urine. So therefore, there must be proteins that are using ATP to actively transport them out of the filtrate and make sure that they appear in very high concentrations in the blood. There are some waste products that may move by either active or passive transport into the filtrate. So we see protons and ammonia, or urea in the case of humans, passively moving at this point into the filtrate from the blood. Other ions that are necessary may move because if they are more concentrated in the filtrate, they will move by passive transport along with water back into the blood. Then, as we move down the loop of Henle, there is a change as you move deeper into the kidney. The kidney tissue itself becomes saltier. So the further you extend down, we're going to see a passive transport by osmosis of water out of the filtrate and into the back into the blood. You can see the loss of water is going to concentrate the urine more, and so that's why you can see that the color as you get down here to the bottom is getting darker. We're going to move salts, and salt is going to be moved first passively as water has been lost, and then actively through ion pumps to ensure that we are not allowing too much salt to be excreted, because again, salt is a necessary ingredient for all of our cells. The second diagram gives a little bit of a different look, but I think is helpful in other ways to be able to show what's happening. First of all, at the top it gives us the locations, the different parts of the kidney nephron, starting with the glomerulus, what's also known as the renal corpuscle, then movement into the Bowmans capsule to the proximal convoluted tubule, so you can see the movement of urea here first into the blood, the transport actively of glucose, ions, water, and amino acids from the filtrate back into the blood, and the permeability of these tubules to water is very high. So water can definitely move into, and in this case, out of the filtrate back into the blood. Now, as it dips down, the numbers here are giving you the relative osmolarity of the kidney tissue surrounding it. So as you get to the saltier medulla of the kidney, there's going to be an additional movement of water out by osmosis, because if the concentration of salt is very high, if it's a hypertonic solution in the surrounding kidney tissue, then water is going to move by osmosis passively from the filtrate into the kidney. And this is happening as we are descending down the loop of Henle. Now as we ascend out of it, the urine, as you can see that number there, has gotten more and more concentrated, and that would mean that water is going to then flood back into the filtrate from the blood, and that would mitigate all of the efforts of the kidney so far to be able to concentrate only the waste products and unnecessary solutes, and not get rid of that necessary water. So as a result, we're going to see that as we ascend back out of the medulla and into the cortex, which is less salty, because we are now in a hypotonic solution, water would move out. So therefore, we don't find aquaporins, which would allow for the movement of water out. So, in other words, the ascending loop of Henle is impermeable to water. Because water is polar, there is no channel available for water to be able to move out of these tubules. As we move now finally to the distal convoluted tubule, we're going to have this reabsorption and secretion is optional depending on circumstances, again, within the body. So while the ascending loop of Henle is impermeable to the movement of water, it is permeable to the movement of salt. And so by first passively and then actively transporting salt in particular out of the filtrate as the urine moves back up the ascending loop of Henle, now at the very end of the nephron, we have reestablished a hypertonic environment. Once we've reestablished that hypertonic environment, now water is going to start to leave again. And as water leaves, more salt is going to be actively transported out. That's going to ensure that the final product, urine, is hyperosmotic, is very, very highly concentrated. Ultimately, it's going to have the minimal amount of water necessary in order to maintain osmolarity within the body, but it's going to be highly concentrated with waste products like urea. This is going to be really important because otherwise we would have to substantially increase our intake of water because there would be so much volume of water leaving our blood through our urine. Well, you can stop there because the physiology of the kidney nephron is the main learning goal for this particular part of the unit. There are a few additional points that I want you to be familiar with. The first is how it is that hormones are used to be able to control the production of urine. ADH, which is the anti-diuretic hormone, is going to be the primary mechanism by which your body is going to control how much urine is produced. When the osmolarity of your blood increases, meaning your blood is becoming more salty, then your body will release ADH. ADH is going to circulate through your blood. It's going to then increase the reabsorption of water. Increase the reabsorption of water. So that means that more water is going to be taken out of the filtrate and brought back into the blood. Ultimately, that means that less urine will be produced. ADH is also going to trigger a behavioral response. It's going to make you thirsty. Because if you're thirsty, then you're going to drink. And as you drink, you're going to help to, by increasing the amount of solvent, lower the osmolarity of the blood. So again, ADH increases permeability increases the permeability of water from the filtrate and into the blood. So the way that it's able to do that is through the primary type of channel protein by which water is going to move from the lumen or the interior of the tubule and allow that to be able to move through the cell, through the duct itself, and back into the kidney and thus into your blood. So aquaporins are going to be the channel proteins present on both sides of the cells that make up those tubules. So as ADH is received within those cells, it's going to increase the number of activated aquaporins. The more aquaporins there are, the more water molecules will be able to move from the interior of the tubule out. Urinalysis is a helpful diagnostic tool. Diagnostic meaning that a urine sample can be taken and analyzed to be able to diagnose when something is wrong. The most obvious of which to be able to detect are UTIs, urinary tract infections, because if bacteria are present in the lining of the excretory system, so within the kidney nephrons, within the ureter, the bladder, or the urethra, then those bacteria are going to convert all of the nitrates in your blood to nitrites. And so the presence of those nitrites is going to signify that bacteria is there. Secondly, the presence of white blood cells is going to be a second key point to show that the body is fighting infections. There's a large variety of kidney diseases, anything from physical damage to inherited diseases in which the kidney is not properly filtering blood. And so the presence of unusual molecules or molecules in unusual amounts are going to be detected during a urine sample. For example, looking for blood cells. There shouldn't be any blood cells in healthy urine. And particularly if there is red blood cells, that shows probably that there is some physical damage because ultrafiltration is supposed to selectively prevent the loss of very large things like blood cells and large proteins. Those are not supposed to get into the filtrate at any point. Therefore, that shows that there has been some damage. The presence of glucose can be important for detecting diabetes. There could have been, again, physical damage showing that for some reason glucose is not being reabsorbed. Or if glucose is present in excessively high amounts in the blood, then there are probably not enough protein channels working to be able to carry those glucose molecules by facilitated diffusion and carrier proteins by active transport from the filtrate back into the blood. Because under normal conditions, we would expect 100% of that glucose to get reabsorbed. Pregnancy tests are also going to be an important tool that ultimately uses the presence of a protein that's acting as a hormone, which is water soluble and thus is carried into the blood. And this protein is only going to be produced during a pregnancy. And so therefore the presence of that particular hormone in the urine is a sign that a pregnancy is occurring. And finally, other unusual proteins could be selectively tested for through drug testing. And so drug testing can be done, for example, for athletes to determine whether or not certain types of banned substances that are soluble in water have actually been taken. Additionally, there are problems with the prescribing of prescription painkillers and those patients that have been prescribed things like opioids, which can be detected through urinalysis. Doctors may require their patients to be able to undergo a regular urinalysis while they're on those opioids to ensure that that patient is taking them and not selling them on the black market because there is a very robust secondary market for opioid painkillers as a way to be able to satiate the habits of addicts. There are issues with urinalysis. It's not a perfect science, and in many cases, in order to ultimately have certainty, further blood testing would need to be done because here we are just looking at the filtrate. We're not really looking at a true picture of what's happening inside the body. And so whether or not urinalysis alone can be conclusive for things like banning an athlete from being able to participate in a particular sports competition is something that should be addressed by a rules committee in order to ensure that all of those actions are ethical. Kidney dialysis is a process that's going to be that a patient will undergo who has some form of kidney failure. Dialysis is a temporary measure. It's very time consuming and expensive, and so therefore it's not a long-term solution. It's really a temporary solution until a patient can recover or a patient who is awaiting a kidney transplant. And finally, an important adaptation to go back to the kangaroo rat, which appeared here earlier in this particular video. The way that the kidney of the kangaroo rat is able to accomplish the production of so little urine is by varying the length of the loop of Henle. The loop of Henle is much longer in kangaroo rat and other desert animals. Desert animals can't afford to excrete a lot of water. Therefore, they need to produce a very small amount of urine, but that urine is going to be relatively more concentrated. And that is illustrated here in this schematic, where you can see the length of the loop of Henle is much shorter in the human versus the kangaroo rat, because it is in that loop of Henle that the process of reabsorption, particularly the reabsorption of water, it's where it's going to occur. So by having a longer loop, more water will be reabsorbed, therefore there's going to be a smaller volume of urine excreted. As always, thank you for watching. Let me know if you have any questions.